Okay, guys, welcome to the lecture over the lymphatic system. As always, I've provided you with a set of learning objectives. I would look over those and think about those as you go through the material. And more importantly than that, if you're in my class at Austin Community College, I would either print out the handout associated with this lecture so you can take notes by hand as you follow along with it, or I would have that handout up and available like in a Word document so you can type your notes as you follow along with it, because this is going to form the basis, for example, of the lecture exam. So when we think about the lymphatic system has three major functions. Major function number one is that at the level of capillaries, capillaries push blood plasma across the capillary wall. That process is called filtration. And what filtration produces at the level of capillaries is a type of fluid called interstitial fluid. Remember that you have way more interstitial fluid than you have blood plasma. So the fluid bathing your cells is called interstitial fluid. If that fluid is allowed to accumulate forever, right, what it will do is it will start accumulating in your tissues, for example, like the tissues that make up your hand, and that fluid will accumulate, producing a condition of edema, which is essentially excess fluid. So we need a way to get that excess interstitial fluid out of those tissues. Function number one of the lymphatic system is it returns excess interstitial fluid back to the cardiovascular system, but first it filters it through lymph nodes, which is really good with respect to our immune function. Number two, the lymphatic system transports digested fats from the small intestines into our system. In other words, there are specialized lymphatic structures that absorb fat in the GI tract. That's one of the functional roles of the lymphatic system. So we have the returning of excess interstitial fluid, we have the absorption of fat in the GI tract, and then we have the production of lymphocytes. And you can think of the lymphatic system as playing a major role in immune responses. So it serves a multitude of roles, the lymphatic system, but one of them is through the production of lymphocytes and through the filtration of things like lymph and blood. It plays an important role in immune response, right? Now, when you think about what I'm talking about with respect to lymph, we have to review our different forms of extracellular fluid. So we've gone over the cardiovascular system at this point, and we know that when the heart contracts, it contracts and it moves blood through a series of blood vessels. The blood vessels that move blood away from the heart are called arteries. And arteries are under high pressure. They branch and branch and branch again, and arteries deliver blood to capillaries. So we call this the arterial end of the capillary here. This is O2 rich, nutrient rich blood. And as blood passes through the capillary, there's a few things I want you to note about the capillary. One, the capillary is so small that red blood cells fit through about one at a time. So the diameter of a red blood cell is about 8 micrometers. So you can't see individual capillaries, but you can see the collective action of capillaries. But individual capillaries are too small to be seen with the unaided eye. You need a microscope. And two, if you look at the capillary wall, right, it's going to be permeable to stuff because it has small gaps or fenestrations in it. And we'll focus on fenestrated capillaries for the moment. So when your heart beats, it creates a blood pressure. It moves blood around your cardiovascular system. So out here is blood plasma. There's some plasma proteins not drawn to scale. Here's some red blood cells. And as blood gets to capillaries, remember capillaries are the most abundant and most important type of blood vessel because that's the point at which exchange occurs. Gas exchange, nutrient exchange, metabolic waste exchange, all exchange occurs at capillaries. In fact, if you think back to the cardiovascular system, if you're thinking about systemic capillary, you can identify capillaries because they go from being red, which indicates oxygen-rich or oxygenated blood, to blue, which indicates oxygen-poor or deoxygenated blood, right? That indicates that exchange has occurred. If a blood vessel changes from red to blue, you know that that has to be a capillary if it's changing right there, because that's the only type of blood vessel that allows for that type of exchange. So we have this capillary running through, and a little bit of excess hydrostatic pressure, which was our P subscript H, forces that blood plasma across the capillary wall. And not everything is moved, like red blood cells and proteins are too big, so that blood plasma gets forced across the capillary wall, and only the plasma and small water-soluble solutes like ions and sugars and amino acids get pushed across. Once that plasma gets pushed across the capillary wall, when it's no longer in the cardiovascular system, it's no longer within the blood vessels, that plasma, right, transitions to become what's called interstitial fluid. Interstitial fluid doesn't have blood cells or blood proteins in it, so it's a little different. Now, that interstitial fluid, which is much more abundant in our body, is what actually surrounds the cells of our tissues. And the process by which plasma becomes interstitial fluid, meaning the process by which we push plasma across the capillary walls in order to form interstitial fluid, that process is called filtration. It's called filtration because we don't push all of those things across. So the red blood cells and the proteins are essentially being filtered out at the capillaries. They're not allowed into the interstitial fluid. So when plasma is pushed across the capillary wall, forming interstitial fluid, that process is called filtration. When interstitial fluid gets reabsorbed back into the capillary, becoming blood plasma again, that process is called reabsorption. And filtration and reabsorption are really points of exchange, right? So we push good stuff like nutrients, signaling molecules, right? Gases are diffusing at this point, and that good stuff goes into the cells, and then that bad stuff comes out of the cells, like the metabolic waste and the carbon dioxide, and most of it gets picked back up by the capillary network. But not all of it. So interstitial fluid continues to build up, and if interstitial fluid is allowed to build up forever, again, it will start to pool, creating edema, and that's problematic because you don't want edema in tissues. So the system that drains excess interstitial fluid, and its primary function is to drain excess interstitial fluid, is the lymphatic system. So here we have an arterial, and we know that these are capillaries because they go from being red to blue. The only type of blood vessel that allows for that to happen are capillaries. So what we're looking at here is what's called microcirculation. You wouldn't be able to see this with the unaided eye. We're talking about the cellular level, right? The micro. You need a microscope. So you have your arterioles, and they branch and they feed into these capillary networks. We know they're capillaries because they go from being red to blue, which tells you about the oxygenation status of those red blood cells. The moment that blood plasma gets pushed across the capillary wall, so the moment that blood plasma gets pushed across the capillary wall, it's no longer called blood plasma, it's called interstitial fluid. So if I was to ask you on an exam, if I was to point here and say, which of the following would be true of the space indicated by the pointer? These spaces here aren't hollow or empty spaces, right? They're filled with things like extracellular matrix, and the type of fluid that allows for the exchange of materials with these cells in their environments is interstitial fluid. Now, the process by which interstitial or blood plasma becomes interstitial fluid, meaning it gets pushed from the cardiovascular system into the interstitial space, that process is called filtration. The process by which interstitial fluid gets reabsorbed back into a capillary to take that material back to our circulatory routes, that process is called reabsorption. And you get filtration and reabsorption happening all the time at the level of capillaries. Now, every blood capillary network you have in your body essentially also has a lymphatic capillary network. Lymphatic capillaries are there to essentially drain excess interstitial fluid. So any excess interstitial fluid will get pushed into this lymphatic capillary. Once that excess interstitial fluid gets pushed into this lymphatic capillary, it's no longer called interstitial fluid. The moment it enters into the lymphatic capillary, it's called lymph. That lymph goes from lymphatic capillaries to lymphatic vessels, and lymph and venous return of blood to the heart will always be going in the same direction. So if you look at a diagram that depicts lymph going in a different direction than venous return of blood to the heart, that's problematic. In other words, lymph is always moving back toward your heart. So if you want to identify the direction of lymph flow and figure out, okay, that's a vein or that's an artery, if the lymphatic vessel is carrying lymph the same direction as the blood vessel, you know that blood vessel is a vein because veins bring blood back to the heart. Venules are just small veins. They'll fuse to form bigger vessels called veins. So that's where lymphatic fluid comes from. And one of the major points in this particular lecture is just being able to describe lymphatic circulation. So you have your blood capillary, you have billions of blood capillaries all over the body. Every single one of your cells essentially is fed by a capillary network with the exception of the cells of the cornea. Now you can see what's called capillary reperfusion when you squeeze on your finger. So if you squeeze on your finger, right, you apply pressure and you essentially clamp down capillaries, and you'll notice your finger goes from being red to being white. And then when you let go, you'll notice that it becomes red again very quickly. That particular phenomenon is called capillary reperfusion. It gives you an idea of how well blood is flowing in that part of the body. If capillary reperfusion is happening very slowly or not at all, it could indicate something like a blood clot feeding to that extremity or that limb, or it could indicate a problem like congestive heart failure, or diabetes can produce situations in which you're not perfusing your limbs with blood very well as a consequence of drops in blood pressure. So capillary reperfusion can actually tell you quite a bit about the way your cardiovascular system is working. So you have this capillary network. We have billions of capillaries. With every blood capillary network, you're also going to have a lymphatic capillary network. So these lymphatic capillaries kind of perfuse through these blood capillaries, and the job of lymphatic capillaries is to drain excess interstitial fluid. So that interstitial fluid moves from the interstitial space into this lymphatic capillary, and this lymphatic capillary will ultimately, bring that lymphatic fluid into lymphatic vessels. So from lymphatic capillaries, those capillaries will fuse over and over again to form lymphatic vessels, and we're going to look at lymphatic vessels. Those lymphatic vessels, which have valves, because lymph is a very low pressure circulatory system, meaning that there's not a heart pumping lymph. The movement of lymph requires things like body movement, gravity. It's not a high pressure system at all. So lymph then moves into these little nodes, these bean-shaped structures called lymph nodes. Lymph nodes filter that lymph for things like pathogens, toxins, and you have bunches of lymph nodes, and eventually, those lymph nodes filter that lymph, filter. After the last lymph node, you get what's called the lymphatic trunk to lymphatic duct, back to the heart. So when you think about the first step in the flow of lymphatic fluid, you have your interstitial space out here. Blood capillaries are around somewhere, and when you look at these lymphatic capillaries, you see these endothelial cells. And these endothelial cells overlap one another to form these flaps. We call them flap-like mini-valves. If the pressure in the interstitial space exceeds the pressure inside the lymphatic capillary, the flap-like mini-valve will open and fluid will flow from the interstitial space into the lymphatic capillary. So the fluid out here would be called interstitial fluid. The moment that fluid, enters into the lymphatic capillary, it's no longer called interstitial fluid, it is called lymph. And then that lymph circulates from the capillaries to the vessels, to the lymph nodes, to the lymphatic trunks, to the lymphatic duct, back to the cardiovascular system. And when lymph is returned back to the cardiovascular system, it's called plasma again. So you have plasma to interstitial fluid to lymph, back to plasma to interstitial fluid to lymph, and that circulation will continue until you die. So when you're looking here we have an arterial, here we have a venual, blood capillaries. These lymphatic capillaries grow with the blood capillaries. You have your interstitial space. In this interstitial space, there's interstitial fluid. If the pressure in this interstitial space exceeds the pressure in the lymphatic capillary, these flap-like mini-valves in the endothelial cells will open. The moment that fluid enters into the lymphatic system, it's no longer called interstitial fluid, it's called lymph. Lymph then circulates through lymph nodes. Those lymph nodes filter that lymphatic fluid, So they filter for things like pathogens and toxins. From the lymph nodes, we get to lymphatic trunks, from lymphatic trunks to lymphatic duct, from lymphatic duct back to the cardiovascular system. So let's trace it one more time. Interstitial fluid, flap-like mini-valve. So we know we're looking at these endothelial cells, and we know we're looking at a lymphatic capillary because that flap-like mini-valve. Pressure out here exceeds pressure in here. Interstitial fluid drains to become lymph. Lymph then circulates, it gets filtered through lymph nodes where it enters into lymphatic trunks and then into lymphatic ducts. And then those lymphatic ducts return that lymph back to the subclavian veins, depending on whether you're talking about the right or left lymphatic duct. The left lymphatic duct is where you get the overwhelming majority of the lymphatic trunk fusing and draining about 85% of your body's lymph. The moment that lymph enters back into the cardiovascular system, it's no longer called lymph, it's called blood plasma. So that's our focal point. So when you think about the primary function of the lymphatic system, it's to drain excess interstitial fluid, preventing edema in body tissues, right? And that's function number one. Function number two is lymphatic system structures absorb fats along the GI tract. So they transport fat or absorb fat along the GI tract. So this guy right here, right, this is a really interesting person. His name was John Hunter, and he was a physician in the 18th century that's often referred to as the father of modern surgery. And he took even though he's kind of a rough and gruff guy, he took a scientific approach to surgery, and many of the surgical procedures we use today and the change in the way we conduct surgery, we owe to Dr. John Hunter. As part of his work, and his work was expansive, I mean, he used like cadavers and things. He had people going to the morgue and to, graveyards to rob bodies. But one of his discoveries was he figured out, at least in part, one of the functional roles of the lymphatic system. And he did that by taking dogs, essentially stray dogs, and he would open them up, and nobody knew what these vessels feeding into the GI tract did. Right? The lymphatic system in the 18th century was largely a mystery. So he would take these dogs, open them up, and go, what do these vessels do? What are these vessels responsible for? And in order to figure that out, he exposed the GI tract to different substances. And one of the substances which has a high amount of fat he exposed the GI tract to, so here's the GI tract, is he dropped milk on it, right? And one of the things he saw with the milk is that the milk fat would be absorbed along the GI tract because that's the job of the GI tract, right? When you think about specifically the small intestines, the job of the small intestines, and we haven't talked about the digestive system, but their primary role is that's where the overwhelming majority of absorption takes place. So when you eat food, you break those macromolecules down into their constituent parts. You have ions, minerals, nutrients, right, water, and you need to absorb that stuff, and you need to get it from the lumen of the GI tract into the blood. So water-soluble compounds like vitamins, minerals, amino acids, sugars, they're absorbed directly into blood capillaries, and then from the blood capillaries, so they go into these blood capillaries, and water-soluble components enter the blood capillaries. They're then drained by veins, and ultimately all of that blood will be taken to the liver via hepatic portal circulation, which we've talked about, and all of that material leaving the GI tract, all of that blood draining from the GI tract, that nutrient-rich blood and sometimes toxin and pathogen-rich blood will first go to the liver. The liver filters and processes that blood, so it uses sugars to make things like glycogen, amino acids, to make all those proteins the liver makes, and then that filtered and processed blood is allowed to enter into your systemic circulation. Fats, lipid-soluble compounds, things that don't dissolve in water, they don't enter into your bloodstream initially. Fat-soluble compounds enter into the lymphatic circulation directly. So you get these absorption of these little things called microns, and we won't talk about that. Just think about it as being fats. And these fatty materials get absorbed directly into the lymphatic system. Specialized lymphatic capillaries that absorb fats along the GI tract are called lacteals. And the reason that they're called lacteals is because John Hunter, when he saw that the milk fat was absorbed not into the bloodstream but into the lymphatic system, he referred to this specialized lymphatic network, or this specialized vessel that was enabled for the absorption of fats, as a lacteal, to pay homage to lactose or milk sugar. So lacteals are specialized lymphatic capillaries that absorb fats along the GI tract. Now those fats enter into lymphatic circulation, not into blood circulation, so they actually bypass what's called first pass liver metabolism. So they don't enter directly into the hepatic portal system. They get emptied essentially into your cardiovascular system, into your subclavian veins, and there's consequences to that. So job number 2 of the lymphatic system is to absorb fats along the GI tract. The specialized lymphatic capillary embedded within these little finger-like projections, which increase surface area for absorption in your small intestines, are called lacteals. Now, job number 1 of the lymphatic system is to drain excess interstitial fluid. Job number 2 of the lymphatic system is to absorb fats along the GI tract. All of those lymphatic capillaries will eventually lead to what are called lymphatic vessels. So lymphatic capillaries are too small to see, but on this lymphatic man and on your image, lymphatic woman, all of these vessels you see out here highlighted in green, all of these vessels are lymphatic vessels. Now you can look through the lab model. We have the video of all the lab models up under the study guides tab. And you'll see there's a model that I refer to as lymphatic man, and you can see the lymphatic vessels really readily. They're white on lymphatic man. That lymph then moves through lymphatic vessels, and those lymphatic vessels deliver that lymph to these bean-shaped structures peppered all throughout your body called lymph nodes. There are 4 beds of lymph nodes I want you to be familiar with. So the lymph nodes that run up the cervical region that you can kind of feel on the posterior aspect of the neck, those are called your cervical lymph nodes. The nodes that come up under your mandible right here, those are called your submandibular lymph nodes. The nodes that filter a lot of the material coming from the upper appendages that you see about right here, right in your armpit region, those are called the axillary lymph nodes. So these guys here are the axillary lymph nodes. And then these nodes down here that kind of project along the groin are called the inguinal lymph nodes. Now, when you think about lymphatic woman, I would be able to label the spleen, the thymus, which sits over the heart, and we'll go back over that in a couple of slides now, and the red bone marrow, because we're going to talk about those. But for this particular slide right here, and what we're going to be discussing now, because we're going to backtrack and discuss those structures later, are the lymphatic flow structures. So in simple terms, lymph flows from lymphatic capillaries. All of that excess interstitial fluid is drained into those lymphatic capillaries. Those lymphatic capillaries fuse to form lymphatic vessels. That lymph moves through the lymphatic vessels. Those lymphatic vessels feed lymph into lymph nodes. Lymph nodes filter lymph for things like pathogens and toxins. And after the last lymph node, so you see here down, we'll look at the lower appendage to highlight this. After the last lymph node, you have lymphatic trunks. Now, you guys don't need to know, for example, the abdominal the intestinal trunks, like the right and left intestinal trunks, but these are called lymphatic trunks, and you have lymphatic trunks peppered all throughout your body. They're not part of your common course objectives. So you have your lymphatic trunks, and the lymphatic trunks from the inferior portion of the body all feed into this localized enlargement where you get this kind of large flow of lymphatic fluid circulating called the cisterna chyli. So this structure right here is called the cisterna chyli. You can see it really readily on the model we're going to be using. Then the cisterna chyli allows lymph to flow into the largest and most important of the lymphatic ducts, and that's called your left lymphatic duct, aka your thoracic duct. So the left lymphatic duct, aka the thoracic duct, in some people essentially drains all the lymph because only 15% of people have a right lymphatic duct. Even if you have a right lymphatic duct, the left lymphatic, aka the thoracic duct, drains 85% of lymph. So it's my focal point usually when I talk about lymphatic circulation. So if you look at this lymphatic duct, the thoracic, aka the left lymphatic duct, it comes up, loops around, fuses to the subclavian vein, and the moment it dumps that lymph back into the subclavian vein, that filtered lymph, right? The moment that lymph exits the lymphatic duct and enters back into the subclavian vein, it's no longer called lymph, it's called blood plasma. The subclavian vein will feed into the brachiocephalic vein, which will ultimately drain into the superior vena cava, which will take blood back to the right side of the heart, where it will enter into the pulmonary and ultimately systemic circulation, back to capillaries, capillaries to interstitial fluid, so plasma at capillaries gets pushed across the capillary wall, becomes interstitial fluid gets drained by lymphatic vessels, becomes lymph, right? Now, the cisterna chyli is a really interesting structure. I was watching a surgery one time. I was actually present in the room and I was scrubbed in and I was in the back of the room and I saw a surgeon accidentally clip a little bit of the cisterna chyli and it just started leaking all over the place. It was unbelievable how much fluid actually came out and they were sitting there sucking up the fluid. It was a really interesting experience. So, that localized enlargement, a lot of lymph is draining in that direction. Now, lymphatic vessels are like veins in the fact that the pressure of lymph flowing through them is very, very low. In fact, it's much lower than veins because there's no heart, right, creating a pressure gradient that forces lymph through the system. So, lymphatic vessels have these really thin walls and they also have what are called valves. And remember, valves from the cardiovascular system prevent the backflow of material. So these lymphatic vessels are very, very thin. They don't have the different tunicas, and they have these valves in order to prevent the backflow of lymph because lymph is under such low pressure. In fact, one of the best ways to facilitate lymphatic return or the return of lymph back to the cardiovascular system and to circulate lymph is to move around. Just the kinetic energy from moving around and squeezing on lymphatic vessels, etc., can actually promote lymphatic circulation. So the saying "movement is medicine" is really a true statement. And I've told you guys about working in the hospital and with the kids who had suffered spinal cord injuries and just getting them out of bed and moving them every day facilitates venous return of blood to the heart, facilitates lymphatic circulation, synovial fluid circulation, prevents the development of things like bed sores and localized edema. Really important. They're not just vegetables once you have a spinal cord injury. You got to move them around, man. You got to circulate those body fluids. Now, metastasis. The lymphatic system becomes really important from a diagnostic perspective. I mean, it's a whoppingly important system, but from a diagnostic perspective, and one of the cases I always like to bring up is the case of metastasis. So you have a primary tumor, and tumors typically have a primary site. For example, breast tumors have a primary site in the breast. With advances in genetic medicine, etc., we'll probably, I think within 50 years, not call breast tumors. We'll call them like P53 mutation tumors or whatever it is, whatever genes are involved, because that gives you more information. But you have your primary tumor, your primary site. And remember, cancerous cells are just cells that are always in mitosis. So they're constantly dividing. They're constantly undergoing the process of mitosis. And because of that, they build these big chunks of non-functional tissue that essentially divert metabolic resources to them, drown out what functional tissues are doing, grow their own blood vessel networks, and they become problematic because they're just chunks of useless tissue, but they're using all these resources. Tumors you can think of as being kind of like dandelion seeds. So if one of those cells sloughs off a tumor, it can enter into the cardiovascular system or it can enter into the lymphatic system. And when it travels through either the cardiovascular system or lymphatic system, you can think of those mitotically active cells as being like little seeds. And then when they implant somewhere, they can start creating distant tumors. When a tumor spreads beyond its primary site or when it advances beyond its primary site, either through the cardiovascular or lymphatic system, we refer to that as metastasis, the spreading of a tumor. Now, Cancer Biology 101, when you're staging cancers, there's stage 1, stage 2, stage 3, stage 4, and the earlier you get it is typically better, and the reason why is because the earlier stages tend to be localized, so surgical removal and maybe some adjunctive chemotherapy is good. So stage 1 cancer, essentially you have a small tumor in one spot. Stage 2 cancer, you have a large tumor in one spot. Stage 3 cancer occurs when cells slough off, enter into the lymphatic system, and you can find those cancerous cells in lymph nodes. So you can start to get the buildup of cancerous tissue in lymph nodes. If cancer has spread to the lymph nodes, it's considered stage 3 cancer. If that cancer has spread beyond the lymph nodes, it's considered stage 4 cancer. So it's important in doing things like staging cancers. Now, the regional lymph nodes you need to know, again, are the cervical, the submandibular, the axillary, and the inguinal. You often palpate or feel lymph nodes to evaluate for the presence of infection. For example, I used to work at an STD clinic, And I would have people come in from time to time who would say, I think I have HIV. I was right next to the University of Arizona, and they'd say, I think I have HIV because one of my lymph nodes is a tiny bit swollen. And I would go, well, that's good. You're reading and you're conscious of those things. I think you have guilty penis syndrome personally. But one of the things that's characteristic with what's called conversion illness with HIV is people develop a rash, a really high fever, and generalized what's called lymphadenopathy. That means generalized swelling of the lymph node. So you don't typically get just a little swelling in one lymph node. The lymphatic circuits in general, because you're fighting a systemic infection, the lymph nodes can get swollen. Same thing when you're fighting the flu, because that's a systemic infection. So I've seen people who've been diagnosed with HIV who've had gigantic lymph nodes. So those lymph nodes can swell in response to infection, and we'll talk mechanistically about how that happens. But generalized lymphadenopathy associated with like HIV, I've seen kind of extreme examples, me personally. So if a clinician is ever just kind of doing a quick and dirty evaluation to see whether you got something like the flu as opposed to something like strep throat, they may feel all your lymph node beds, right? And if you got inflammation or those lymph nodes are swollen systemically, you might have a systemic infection like the flu. Or if those lymph nodes are swollen locally, like the submandibular lymph nodes, it might be a localized infection like strep throat. So, when you're looking at a lymph node, you don't have to know all of the histology of the lymph node that, for example, other classes do. In fact, we'll go over the histology of the lymph node you need to know. So lymph nodes are these kind of bean-shaped structures. And one of the things I want to highlight about lymph nodes is they're surrounded by a layer of dense connective tissue. And that layer of dense connective tissue surrounding the lymph node is called the capsule. So lymph nodes are true organs because they have this capsule on them. They're not lymphoid organs, they're lymphatic organs. Not a major point, but worth mentioning. So you have this dense connective tissue capsule. Now, lymphatic vessels, afferent vessels, bring lymph to the lymph node. That lymph then enters into what are called the subcapsular sinuses. So all these little hollow spaces through which lymph is flowing, that's a subcapsular sinus because it's directly deep to the capsule. Then you get the trabecular sinuses because these connective tissue networks that fold in are called trabeculae, and these trabecular sinuses allow that lymph to travel internally in the lymph node. In the center of the lymph node, you have the medullary sinuses, which feed back into the trabecular and ultimately the subcapsular sinuses into the efferent lymphatic vessels. And after the last lymph node, you're no longer talking about lymphatic vessels, you're talking about lymphatic trunks. Now, as you're going through this, when you look at lymph nodes, remember that if pathogens, right, are exposed to one of these lymphatic nodules, the job of lymph nodes is to filter for things like, toxins and pathogens. Now, you don't have to know all of these different cell types. If it was anatomy and physiology 2, I would make you know it. For the intro class, I don't make you know it. But you have these lymphatic nodules, and the germinal center of a lymphatic nodule is essentially an activated lymphatic nodule that contains a constellation of white blood cells. And as things like pathogens that these white blood cells are responsible for targeting pass next to these nodules, these lymphatic nodules, those white blood cells target those pathogens for destruction, either using the cells themselves or antibodies. And the lymph that comes out the other end of a lymph node, right, has a lower pathogen toxin count than the lymph that came in. So they're filtering mechanisms. They're filtering lymph before it's allowed to return to your cardiovascular system. And in that respect, the lymphatic system plays an important role in immune responses. Another thing that can happen is, let's say you have a infection that you're fighting, and in response to that infection, the white blood cells in your body start to divide, so there are more of those types of white blood cells available to fight that infection, right? As those cells divide, they physically take up more space in the lymphatic nodule, so it's the division of white blood cells in response to infection that actually causes the lymph node to grow, right? And you can actually see that, you can feel it, and you can have prolonged enlargements of the lymph node. You call them a rubbery lymph node once that pathogen is passed. So that's what causes lymph nodes to swell. So When you look at a histological image of a lymph node, here we have the capsule, the subcapsular sinus. Here we have the lymphatic nodules. And when you look at each of these, the kind of white centers of the lymphatic nodules, those are called germinal centers of lymphatic nodules. And as the lymph flows past those lymphatic nodules, the white blood cells in those lymphatic nodules target pathogens and toxins and destroy them. So the lymph that comes out of the lymph node, right, has a lower pathogen toxin count than the lymph that entered the lymph node. Now, there are some complexities there that we can layer on that we're not really going to talk about in this class. So when you think about swollen lymph nodes, I have seen, these are the cervical lymph nodes right here, but I have seen cervical lymph nodes, submandibular lymph nodes that have been like the size of grapefruits. I mean, they can become really swollen depending on the nature and severity of the infection. Now, after lymph nodes, lymph enters into lymphatic trunks. So you have the intestinal trunk and the lumbar trunks. And those lymphatic trunks, you don't have to know the intestinal and lumbar trunks, as I stated earlier, but those lymphatic trunks, right, bring that lymph to this localized enlargement called the cisterna chyli, which you can see really clearly on the model. From the cisterna chyli to the left lymphatic, aka the thoracic duct. That left lymphatic duct then brings that lymph back and it fuses with the left subclavian vein. Subclavian vein runs directly deep to the clavicle, and once it fuses with the subclavian vein and it dumps that lymph into the cardiovascular system, it's no longer called lymph, it's called blood plasma again. That blood is then returned back to the right side of the heart where it enters the respective circulations that we talked about before. But remember, it's undergone a filtration process. So that lymph returning back has been filtered through your lymphatic systems, and that's an important part of your immune response. It's one of the reasons that it's good to drink a lot of fluids when you're sick, because you increase the flow of fluids, like the circulation of fluids in your body, and you increase the number of times that those fluids are filtered through the lymphatic systems, and that's a good thing. So here we have the right lymphatic duct. Again, not my focal point for something like the exam. It's not that it's unimportant, it's just that only a small percentage or a relatively small percentage of people have it, and the left lymphatic duct plays such an important role with respect to the volume of lymph that it drains is that I draw a focal point to that one. So left drainage area, right drainage area, that's still important for people who have a right lymphatic duct. For example, some women who get mastectomies can develop a condition in which, some of those lymphatic vessels are damaged. And if you damage the lymphatic circulation, you can develop severe edema, for example, in your arms. A mastectomy is a surgical removal of a breast, by the way. So the final function of the lymphatic system is it facilitates, to some degree, immune responses. It's not the only thing, but it contributes to immune response. So in order to understand the immune system, essentially what your immune system does is it distinguishes self from non-self, right? So anything that's considered self, and this is when we talked in the first unit about the major histocompatibility complexes, the immune system leaves alone. Anything that's considered non-self, the immune system targets for destruction and destroys. Now, the immune system itself, if you're looking at your follow-alongs, there's two tables that say innate immunity, right, and adaptive immunity, or non-specific and specific immunity. So two different words meaning the same things. Innate immunity, right, consists of defenses that are present at birth that provide rapid response to an array of pathogens. So defenses that are present at birth that provide a rapid response to an array of pathogens. These include physical barriers. So for example, your skin contributes to innate immunity just by preventing pathogens from being able to enter into your system. The mucous membranes in your lungs, the mucous membranes that line like the reproductive and the urinary tract, the digestive tract, they prevent the infiltration of pathogens, but just by forming a barrier. You have secretions like saliva, like mucus along the GI tract, like mucus in the mucociliary clearance of your lungs that just trap pathogens and move them out of your body. You have cells that undergo phagocytosis that just go around and gobble things up. But one of the important things about innate or non-specific immunity is that they will respond to any immune threat, right? Meaning that they'll be present with any immune threat, and they don't form a memory. So innate or non-specific immune responses don't form a memory, and they'll target every pathogen the same way. Adaptive or specific immunity, which is often what we think of when we talk about our immune system, is lymphocyte-mediated immunity. And lymphocytes are different than other types of white blood cells in the fact that they recognize specific pathogens and they launch specific responses to specific pathogens. Also, within that immune response, those lymphocytes will create bundles of cells or groups of cells called memory cells. And those memory cells will stick around, and if you're ever infected with that same pathogen again, those memory cells will recognize it and you'll launch a massive immune response. So when you get something like a vaccination, what you're playing around with there is not innate immunity. What you're doing is you're cueing adaptive or specific immunity, and you're forming memories, right? Because those cells can form memories. So if you put a deactivated form of a virus in and that triggers a small immune response, right? That virus, like let's say smallpox, right? That virus that's not harmful to you, the deactivated form of it, can still trigger your immune system, and you can still have those memory cells being formed, and it's those memory cells that will prevent you from actually getting smallpox, for example. So adaptive or specific immunity is entirely lymphocyte mediated, and it's a specific, they recognize specific pathogens, launch specific responses, and they form memories of pathogens you've had in the past. So external immune defenses, things like your skin, the secretions coming from different parts of your body that protect like the mucous membranes in your lungs, different antimicrobial enzymes, urine in your GI tract, that's an innate or non-specific immune defense. I often tell students, hey, when you're done having sexual intercourse, it's not a terrible idea to urinate. It's even a better idea to use protection, especially if you don't know the person. So you don't have to know all of those different proteins and responses for the introductory class, but suffice it to say, there are a lot of different innate or non-specific immune defenses. Specific immune responses are a little bit different. Now, what you should be looking at is an image that looks like this. Your two primary organs of the immune system, so the two primary organs that compose your immune system, which is a little bit different than the lymphatic system. The lymphatic system is a component of the immune system. But the two primary organs that compose your immune system are the thymus and the red bone marrow. So why would red bone marrow be considered a primary organ of immunity? It's because red bone marrow is capable of producing red blood cells, white blood cells, and platelets. If you heard that correctly, red bone marrow is where white blood cells are produced. And white blood cells, right, play an exceedingly important role in both nonspecific and specific immunity. Essentially, red bone marrow is where your immune system derives from. Anything that compromises that red bone marrow is going to be problematic. Now, some of those white blood cells, specifically a certain type of lymphocyte, aren't considered what's called immunocompetent, meaning they can't target infection, and those particular lymphocytes can't target infection until they go to the thymus. Within the thymus, those lymphocytes become immunocompetent. It's almost like lymphocytes going to school to learn how to fight infections at a molecular level, and after they leave the thymus, they can populate different lymphatic structures throughout the body, but at that point, they're considered immunocompetent. So the red bone marrow is considered a primary organ of immunity because it produces white blood cells, and the thymus is considered a primary organ of immunity because it's where a very important branch of those white blood cells goes to mature before those white blood cells can go around your body and target infection effectively. So when you are looking at this particular image right here, you have your red bone marrow. Your red bone marrow is responsible for making red blood cells, white blood cells, and platelets. Specific to adaptive or specific immunity, right? The red bone marrow makes lymphocytes. Now, B lymphocytes migrate directly to the tissues where they're going to work. So you have B lymphocytes, and those B lymphocytes migrate directly to the tissues where they're going to work. The job of B lymphocytes is when they come into a pathogen that they're capable of interacting with, they form right? Plasma cells, and these B lymphocytes differentiate into plasma cells, and these plasma cells produce antibodies. And remember, antibodies are those proteins that float around in your blood that can, for example, tag cells for destruction or bind to viruses or toxins, deactivating them directly, or they can cause agglutination and they can clump together immunological threats. So this is called antibody-mediated immunity, and it's directed against pathogens that are floating around outside of the cell, right? So things like bacterial particles or viral particles. Now, where the thymus becomes involved is the red bone marrow produces lymphocytes. You have B lymphocytes. Notice that when it comes into contact with an infection, it creates a small subpopulation of memory cells. So both antibody mediated immunity and cell mediated immunity, you have a memory component to them, and it's a specific response. So these memory cells, these particular B lymphocytes floating around, will only target that specific pathogen again. And that's why it's called adaptive, because you form a memory, and those memory cells can stick around for a long time, and specific because those memory cells, right, and those particular white blood cells target only specific pathogens. So you have lots of different populations of them. Red bone marrow also produces T lymphocytes. T lymphocytes migrate to the thymus, and in the thymus, they're pinned with these proteins called antigen receptors, right? So they're not able to target infection until they get pinned with these antigen receptors. Remember, an antigen is anything that can trigger an immune response. So the way that the T cell recognizes an antigen is with these antigen receptors. So before that, T cells can't target infection. After they mature and get pinned with antigens in the thymus, they can target infection. Now, if these T cells come into contact with something that they're capable of targeting for destruction, they will activate, and they will also form memory cells. So they'll activate, and those active cells will actually go and mediate the immune response that they mediate, but they'll also form memory cells. So if you ever become infected with that particular pathogen again, your body will know how to deal with it. These active cytotoxic T cells, these T lymphocytes, then attack invading pathogens, but they do so by destroying cells. So once a virus, let's say like a coronavirus, infects a human cell, right? Once that virus infects that cell, and you can go back to the white blood cells where we are looking at lymphocytes, these T lymphocytes don't destroy extracellular pathogens, they destroy intracellular pathogens. So once one of your cells, like let's say a cell in your throat or in your lungs, becomes infected with a virus, once these specific populations of T lymphocyte recognize it, they'll kill the cell. They'll kill the infected cell. If you have a cell become cancerous, they'll kill those cancerous cells. So they kill cells, whereas these guys destroy things outside of cells. These guys destroy the cells themselves. And they work together. So primary organs of immunity are your red bone marrow and your thymus. Now, lymphoid organs are called lymphoid organs because they don't have a capsule surrounding them, so they're kind of these soft lymphatic tissue structures perfused with white blood cells. Lymphoid organs, for you to know, are the tonsils in the pharyngeal region, the thymus, the spleen, the mucosal associated lymphatic tissue or MALT tissue lining the GI tract in these lymphatic nodules, and the appendix. Now, we're going to draw a focal point to just some of the lymphatic follicles or the lymphatic nodules. This is also called mucosa-associated lymphoid tissue. Along your GI tract, a huge percentage of your immunity actually derives from your GI tract, and it's because you have all these bundles of lymphatic tissue lining your GI tract that we'll just call MALT. And when you eat something, right, like when you eat an apple, you don't realize there's 10 to the 8th bacteria on every bite of that apple, or probably more. I'm not a microbiologist, but you're ingesting a huge amount of potential threats to your body. So when you absorb, right, these things, sometimes things like bacteria or viral pathogens or toxins encounter these lymphatic nodules, and that's what allows you to mount an immune defense. So a lot of your immunity actually derives from your GI tract. Just know MALT is that lymphoid tissue that lines your GI tract that plays a really important role in immunity. Now, The other lymphoid tissue I want you to know, one is the tonsils. Now, the most important tonsil with respect to just evaluating somebody's health are the tonsils you can see in the back of the throat, and those are called the palatine tonsils. And then the final lymphoid organ I want you to know is the spleen. When you look at the spleen is in the left upper quadrant. We saw them when we did the cat dissections. We saw the spleen. And the spleen isn't surrounded by a capsule. So lymph nodes are surrounded by that connective tissue capsule. The spleen is actually a soft tissue organ. So if you get an infection and your spleen swells, it's really easy to rupture the spleen because that tissue within the spleen is so soft, right? Even though it's being stabilized by those different connective tissues, that splenic tissue is really, really soft, right? So you have to be careful with it because getting hit there can rupture that and cause massive problems, especially if you have an infection. Histologically, the tissue of the spleen is broken into two different histological tissues that we're going to look at. The first is red pulp. The job of red pulp is to filter old, worn out red blood cells out of the cardiovascular system. So as red blood cells flow through the cardiovascular system, they get old and worn out, and the capillary networks within the spleen are capable of beginning the filtering process to get rid of old, worn out red blood cells. And they don't just get rid of old, worn out red blood cells. The red pulp of the spleen also functions as a reservoir for red blood cells. So you can have red blood cells, right, just hanging out in the spleen. And then if you, for example, increase your physical activity, the cells in the spleen can, right, the smooth muscle in the spleen can contract in order to pump some more red blood cells into your circulation to increase oxygen carrying capabilities. So, if the spleen plays an important role in filtering out old, worn out red blood cells, why would you expect to see an elevated hematocrit when you have a splenectomy or the surgical removal of a spleen? Another thing the spleen does is it has these regions called white pulp, and these white pulp areas are richly perfused with white blood cells. As the sinusoidal capillaries of the spleen allow blood to percolate through the spleen, so the lymph nodes filter lymph, by the way, the spleen filters blood. You have blood coming in, blood coming out. The spleen filters blood. And these white pulp areas, the spleen filters blood, and it plays a particularly important role in filtering blood for bacterial infections. So as bacterial organisms come into contact with the white blood cells composing this white pulp, right, they're engaged immunologically and a lot of times destroyed. So the blood leaving the spleen, so you have blood coming into the spleen and blood leaving the spleen. The blood leaving the spleen, again, has a lower pathogen count. The spleen plays a particularly important role in targeting things like bacterial infections. So that's it for today. That's the intro lecture to kind of give you an idea. Thanks